A while back I did a study on cloning, researching rather. Right. The study was done by Dr. Kelly Hollowell, Institute for Creation Research. Glad for this article. Redefinition, cloning, of when life begins, exposing flaws in the pre-embryo-embryo distinction. People like to make nuances out of something that actually aren't nuances, but editorialisms, fabrications. Cloning is a technological breakthrough. Back in 1999, this was 22 years ago, that adds a new dimension to reproductive technology, as well as new insight into the long-standing debate regarding fetal interests. You can use that word. Currently, a flawed understanding of both science and the law has created a policy that allows for the destruction of countless human embryos created through advancements in reproductive technology, including human cloning. This policy resulted when recent advances in reproductive technology collided with the law, local laws, federal laws, state laws, in the Tennessee Supreme Court, in this case, Tennessee, state of Tennessee, of Davis versus Davis. In Davis, let me get up closer here. The glare, take away the glare. In Davis, a dispute regarding the custody of frozen embryos arose between a husband and a wife, hence Davis versus Davis, who, after undergoing an in vitro fertilization procedure, could no longer agree on the disposition of their frozen embryos. To define the interest that the litigants held in the embryos, the Tennessee Supreme Court relied on a report published by the Ethics Committee of the American Fertility Society. Remember, according to the Bible, these embryos are persons because at conception. In this report, the Ethics Committee defined an embryo as distinct from a pre-embryo based on medical science and legal precedents. According to the report, the pre-embryo stage is considered to last until 14 days after fertilization. Moreover, the consensus concerning pre-embryo status is that the pre-embryo deserves greater respect than that accorded to mere human tissue because of its potential to become a person, but not the respect accorded to actual persons. The Davis Court agreed with the committee report, holding that pre-embryos are not strictly speaking either persons or property, but according to an interim category that entitles them to special respect because of their potential for life, as in Davis versus Davis. So as a result of this decision, our understanding of natural as well as non-coital reproduction now includes a pre-embryo-embryo distinction and a policy that has been defined by the medical community and sanctioned by the courts. This distinction in policy will likely apply to all new techniques for non-coital reproduction, including human cloning. In the process of cloning, the 23 chromosomes of a recipient egg are removed. So I apologize for the angle of this camera, but it's awful glary on the screen today. Any case, similarly, the DNA, or genetic material comprising 46 chromosomes, is removed from a selected adult cell. The 46 chromosomes of the adult cell are introduced into the new, empty, and nucleated egg. I'm getting some new words here. Alternatively, the adult cell is fused with the egg to introduce the 46 chromosomes into it. The egg then contains the 46 chromosomes of the adult cell, and we use the information encoded in the DNA to create a clone of the donor. The 46 chromosomes introduced into the egg are identical to the genetic material contained by all the other adult cells of the donor that contains 46 chromosomes. The genetic material taken from the donor was originally determined, presumably years earlier, when an egg and a sperm each donated their original 23 chromosomes at the point of conception. In natural conception, 23 chromosomes of the sperm and egg unite to create a single cell containing 46 chromosomes. Therefore, the moment that 46 chromosomes are introduced into the enucleated egg is equivalent to the naturally occurring point of conception. In cloning, the life created will be genetically identical to the donor, as though it were an identical twin of the donor. During the development, in both the naturally conceived and the clonal embryos, repeated divisions of the embryo continue to increase the number of cells until they begin to specialize and organize into an adult. Specialization during development is called differentiation. Differentiation is a continual process. Specifically, as the cells multiply and divide, groups of cells become gradually committed to particular patterns of gene activity. Differentiation does not mean that cells lose genes during development. In fact, all differentiated adult cells of an individual are genetically identical. They are simply not metabolically identical. This means that different genes are activated to make proteins as required by the individual cell. For example, the proteins required by liver cells are not necessarily the same proteins required by hair cells. That is why each cell makes different proteins suited to its needs, while the genetic material remains constant in each cell. This explains why genetic material can be taken, theoretically, from any cell and injected it into an enucleated egg, resulting in a clone, typed it in twice, in a clone of the animal or person from whom the cell was taken. In short, since all succeeding cells are genetically identical to the fertilized egg, the magic occurs when a complete set of 46 chromosomes is introduced into an egg, whether by natural conception, in vitro fertilization, or cloning technology. Differentiation is of particular interest because the Ethics Committee and the Davis Court used this developmental phenomenon as a foundational part of the PREEMBRYO-EMBRYO distinction. Specifically, the report and reasoning of the committee make the PREEMBRYO-EMBRYO distinction based on differentiation, which is explained in terms of development of an individual and uterine implantation. The question then is whether this PREEMBRYO-EMBRYO distinction based on differentiation remains valid in the light of the newest reproductive technology, cloning. To make such a determination, a closer examination of the report by the Ethics Committee of the American Fertility Society is warranted. The Ethics Committee explains differentiation in terms of development of an individual as correlated with visually recognizable features and structures of the developing embryo and described in terms related to twining, the splitting of an embryo into two identical twins. Specifically, the committee reports that with the appearance of the primitive streak, as far as is known, the embryonic disc is committed to forming a single being. Beyond this point, twinning is not believed to occur, either naturally or experimentally. Therefore, absent specific visually recognizable structures that indicate an end to the embryo's ability to create a twin, a human embryo is not a person, nor is it properly. It is a pre-embryo. There are at least two recognizable flaws in relying on this explanation as a basis for defining the pre-embryo status. First, while it is conceded that prior to 14 days, single embryos have the ability to split or be split to affect development of more than one independent adult, each life is so created develops in exactly the same manner as the embryo from which it was split. This is the result of being derived from the exact same genetic material. This event merely serves to reset the biological clock of the embryo, forcing it to repeat previously experienced divisions. In humans, this event does not prevent the embryo from attaining eventual personhood. So let me go back and repeat this. First, while it is conceded that prior to 14 days, single embryos have the ability to split or be split to affect development of more than one independent adult, each life so created develops in exactly the same manner as the embryo from which it was split. This is the result of being derived from the exact same genetic material. This event merely serves to reset the biological clock of the embryo, forcing it to repeat previously experienced divisions. In humans, this event does not prevent the embryo from attaining eventual personhood. At a minimum, the embryo will develop into at least one life. It is questionable, therefore, whether the phenomenal ability of the embryo, under some conditions, to produce more than one life should diminish an embryo's life status. Second, evidence that the embryo is a specific life from the moment of conception is actually offered by cloning technology. The moment that a complete set of 46 chromosomes is introduced into an enucleated egg, the embryo is a very specific life, identical to the donor of the genetic material. To illustrate, the success of Dolly and various other cloned animals provides undeniable evidence that the embryo is set on a predetermined pathway of life from the moment the complete set of chromosomes is introduced into the egg. That is precisely the science and logic that explains how the clonal embryo is capable of duplicating the donor. Well, this is tough stuff to understand, so to repeat, the moment that a complete set of 46 chromosomes is introduced into an enucleated egg, the embryo is a very specific life, identical to the donor of the genetic material. To illustrate, the success of Dolly and various other cloned animals provides undeniable evidence that the embryo is set on a predetermined pathway of life from the moment the complete set of chromosomes is introduced into the egg. That is precisely the science and logic that explains how the clonal embryo is capable of duplicating the donor. The individual cells of the clonal embryo, clearly early in cleavage, follow the exact path of development followed by the donor of the genetic material when the donor was embryo. In other words, there appear to have been no options for the clonal embryo as a whole in its development. Therefore, we can infer that there were not options of the initial groups of cells, the pre-embryo, that came into existence through cell division in the first few days of life. Recalling that the moment that a complete set of chromosomes is introduced into the egg is equivalent to the point of conception, it is clear that development of the individual is encoded in the genetic material itself and does not require 14 days to be committed to forming an individual being. Additionally, differentiation is explained by the committee report in terms of uterine implantation. The report states that it is the physiologic interaction of the embryo with the mother during implantation that determines the path of differentiation. Clearly, cloning suggests otherwise. Specifically, the clonal embryo develops in exactly the same manner as the donor, despite the absence of the same available womb. cloned animals, such as dolly